is gluconeogenesis. So we'll see the objective of this class is to understand the role of liver in glucose homeostasis, Cori cycle, glucosalanine cycle, gluconeogenetic pathway, synthesis of glucose from lactate glycerol alanine, and regulation of gluconeogenesis. So these are the things we are going to learn. So first of all, what is gluconeogenesis? So is a biosynthesis of new glucose from non-glucose precursors. So normally what happens, the glucose when it is required for the cell, so usually it takes from the, it's a polymers. So like either some kind of glycogen or some other starch or other polymers like or um, disaccharides or polysaccharides it will take. But if you don't have enough of those and you need to maintain the concentration of the glucose inside the cell and the body and the blood, so that is very important, need to maintain the glucose. So concentration, so gluconeogenesis is plays very important role. Thus it will make it to synthesize the glucose from other precursors, non-glucose precursors. So one of the way is reverse of glycolysis. This is the glycolysis which we have seen from glucose until the formation of the pyruvate, and pyruvate enters the citric acid cycle or pyruvate convert into lactate. This is what you can see the left hand side, all the glycolysis pathway. And this right hand side, what you can see the gluconeogenesis pathway. So almost many steps are in the same as glycolysis but a reverse direction, and the same enzymes except a few where it is has been highlighted here. Glucokinase, which is needed for the glucose to glucose 6 phosphate, is not going to be catalyze the reverse reaction. You need another enzyme. Here also it is needed another enzyme, fructose 6 to fructose 1,6 phosphate, and phosphoenol pyruvate to pyruvate. In fact, pyruvate to phosphoenol pyruvate directly cannot happen, so it will indirectly pyruvate to oxaloacetate to another substrates, and through the some intermediates will enter into the cytoplasm, and oxaloacetate convert into phosphoenol pyruvate. Once it forms a phosphoenol pyruvate, is all the reactions are same until here. Then another, this is the unique reaction to convert into fructose 6 phosphate, and then here another unique enzyme to convert into glucose. So we are going to see especially those steps which are different from the glycolysis, and that is the gluconeogenesis. So what is the importance of this gluconeogenesis? The production of glucose from other metabolites is necessary for the use of a fuel source by brain, testis, lens, cornea, erythrocytes, and kidney medulla, since glucose is a sole energy for the source of the organs. And you will understand other metabolisms like um, fatty acid or protein metabolism in the next classes, where you also get some energy, and cells will get energy. But the problem is some of the tissues like mentioned here are solely depend on the glucose as energy source. The remaining cells are fine. If even though glucose is less, those cells still can survive because it will find the another metabolism as a source of energy. But these cells, especially the brain and the erythrocytes, these are all required mainly the glucose as energy source. But that's why the body need to maintain the glucose concentration constantly. So thus the gluconeogenesis plays a important role to make the glucose synthesis, which are needed for the those tissues. And where the gluconeogenesis occur. So we have seen the glycolysis, right? Glycolysis occurs all parts of the body, every cell plays a role, though liver cells is plays more, but everywhere, every cell of the body, glycolysis occur. And subcellular part, where it occurs, glycolysis, mostly, and not mostly, all reactions are in cytoplasm. But gluconeogenesis are mainly the liver and kidney only, gluconeogenesis. 90% of the gluconeogenesis is the liver, and remaining 10% is the kidney. And subcellular part, partially in mitochondria, very few reactions, and mostly in cytoplasm. And this is the key reactions of the gluconeogenesis. So let us come from the reverse direction of the glycolysis. What happens? Pyruvate is the end product of the glycolysis, right? And not only the end product of the glycolysis, even some of the amino acids, some of alanine, even the lactate can be reverted back to pyruvate. And this pyruvate will be used to synthesize glucose. How it will be, pyruvate cannot be directly converted into phosphoenol pyruvate, which you can see in glycolysis, phosphoenol pyruvate directly pyruvate, but not the case in case of reverse reaction. Pyruvate should be converted into first oxaloacetate with the enzyme pyruvate carboxylase, and oxaloacetate will convert into phosphoenol pyruvate by the help of phosphoenol pyruvate carboxylase. Then phosphoenol pyruvate, the further steps will be convert into glycerol E3 phosphate, and then it will become fructose 1,6 phosphate, and so on. So we'll try to understand these steps first, how this pyruvate convert into phosphoenol pyruvate, and remaining steps we will see later. And okay, enzymes of gluconeogenesis. The synthesis of glucose from pyruvate utilizes many of the enzymes same as glycolysis. Just now I mentioned to you. Three enzymes of glycolysis have such a large negative standard Gibbs free energy change, okay, in the forward direction, that they are essentially irreversible. When the standard Gibbs free energy change is very negative, so a lot of energy is giving during that time. So how it will be revert back? It's not so easy. So that's why even the ATP cannot help you this reaction. So it is irreversible. So these steps must be bypassed in gluconeogenesis. So as mentioned in this previous slide, so these steps, the pyruvate to pyruvate, phosphoenol pyruvate, fructose 1,6 bisphosphate to fructose 6 phosphate, glucose 6 phosphate to glucose, where it required a different strategy than glycolysis. Remaining all steps are just like glycolysis. What are these three different enzymes? Pyruvate kinase, where in glycolysis convert into pyruvate, phosphoenol pyruvate to pyruvate, phosphofructokinase, where fructose 6 phosphate to fructose 1,6 bisphosphate, glucokinase, where it will convert glucose to glucose 6 phosphate in glycolysis. But these three enzymes cannot be used in the gluconeogenesis. What is the alternative? Okay, the first one, pyruvate. How pyruvate can be converted into phosphoenol pyruvate? So in case of glycolysis, a single step, pyrophosphoenol pyruvate to pyruvate. This is a pyruvate synthase enzyme, but pyruvate synthase enzyme cannot be utilized here. So what happens to this? Pyruvate, first of all, whichever present in the cytoplasm, need to be entered into the mitochondrial matrix. That is the first step. Okay. So what is the way? As mentioned in the citric acid cycle, uh, pyruvate cannot directly, I mean, any other molecule cannot go directly from cytoplasm to mitochondrial matrix. So, but pyruvate is having some special membrane protein for transporting that is called as pyruvate translocase. So here the pyruvate translocase allows the pyruvate to enter into the mitochondrial matrix. That is the first step. And what is the next step? Pyruvate need to be converted into oxaloacetate. This is a different step, not seen in anywhere before, right? Pyruvate need to be converted into oxaloacetate directly. So with the help of pyruvate decarboxylase, so where the carbon dioxide will be added to the pyruvate and oxaloacetate will be formed. Pyruvate is a 3 carbon containing molecule and oxaloacetate 4 carbon. So thus carbon dioxide need to be added, ATP utilized, and biotin is a cofactor. Pyruvate carboxylase is an enzyme catalyzing this reaction. So oxaloacetate is formed. So this oxaloacetate need to be transported back to the cytoplasm. But oxaloacetate as it is cannot enter into the cytoplasm. So, but it will go indirectly. How? Oxaloacetate convert into aspartate. Aspartate is having some transport protein and the mitochondrial matrix. So aspartate can be transported back to the cytoplasm. There, cytoplasm aspartate is given rise back the oxaloacetate. So oxaloacetate need to be transported into the cytoplasm, but cannot. It is not having its own transport protein. So indirectly, one of the way is through the aspartate, it will go and again convert back to oxaloacetate. The second possibility is oxaloacetate convert into malate utilizing the NADH, and malate is having the transport protein in the mitochondrial matrix, and then malate enter into the cytoplasm, and malate it convert into oxaloacetate by releasing back the NADH which is utilizing. So energetically there is no much change, but it will pass through the malate or it will pass through the aspartate, and oxaloacetate will be entering into the cytoplasm. This oxaloacetate convert to the pyruvate, phosphoenol pyruvate by releasing the carbon dioxide and utilizing one another GTP. See, one ATP utilized here and one GTP utilized here. But no need to worry about the NADH because whatever the NADH utilized here is released back here. So the net utilization of the energy is one at the conversion of at the step of conversion of pyruvate to oxaloacetate in the form of ATP. Another one is oxaloacetate to phosphoenol pyruvate in the form of GTP. So pyruvate to phosphoenol pyruvate conversion needed these many steps. First need to be enter into the mitochondrial matrix, then oxaloacetate, and then aspartate, then transport aspartate to the cytoplasm, convert aspartate to oxaloacetate, and oxaloacetate to phosphoenol pyruvate. Alternatively, pyruvate enter into the mitochondrial matrix, pyruvate to oxaloacetate, oxaloacetate convert into malate. Malate is having the transport protein to transport to the cytoplasm. Malate convert to oxaloacetate, oxaloacetate to phosphoenol pyruvate. So this one step, pyruvate kinase, which is catalyzing in the in the glycolysis pathway, is a single direction, phosphoenol pyruvate to pyruvate. So in order to make it into revert, it need to perform these many steps. And so pyruvate also not only produced by the phosphoenol pyruvate to pyruvate, also alanine amino acid also will gives the pyruvate. And lactate also will be reversed back to form pyruvate and where NADH is produced. Okay. So that is the step how pyruvate will be converted into phosphoenol pyruvate. So next what happens? So yeah, these are the step-by-step just given. Pyruvate to oxaloacetate, what is the enzyme, and biotin is the cofactor, carbon dioxide is added, and then ATP is utilized. And oxaloacetate to phosphoenol pyruvate in the cytoplasm, phosphoenol pyruvate carboxy kinase, where GTP utilized, and malate to oxaloacetate and aspartate to oxaloacetate. Okay, so here I mentioned to you two ways, right? How oxaloacetate um, can transport back to cytoplasm. But there is another step. What is the other step? This oxaloacetate directly convert back to phosphoenol pyruvate in the mitochondrial matrix. No need to transport oxaloacetate transport to the cytoplasm. Mitochondria is also having enzyme phosphoenol pyruvate carboxy kinase. So there are two enzymes are there. One enzyme present in the cytoplasm where oxaloacetate need to be transported to cytoplasm, oxaloacetate convert into phosphoenol pyruvate in the cytoplasm, or oxaloacetate directly can convert into phosphoenol pyruvate in the cytoplasm, that I will show you in the here. See this pyruvate in this mitochondrial matrix, it will convert into oxaloacetate, and oxaloacetate convert into phosphoenol pyruvate in the mitochondrial matrix itself. So for that, it required the same enzyme phosphoenol pyruvate carboxy kinase, but this enzyme it is different from the cytosolic phosphoenol pyruvate carboxy kinase. So this cytoplasmic phosphoenol pyruvate carboxy kinase coded by the nuclear genes, the genes in the nucleus. But this enzyme coming from the mitochondrial genome. Okay, so mitochondrial phosphoenol pyruvate carboxy kinase is converting oxaloacetate into phosphoenol pyruvate, and phosphoenol pyruvate is translocated to the cytoplasm with its own transporter protein. Okay, so that is the another way of how the pyruvate, the oxaloacetate is converting into phosphoenol pyruvate. Okay, so phosphoenol pyruvate, once it is formed in the cytoplasm, and phosphoenol pyruvate until fructose 1,6 bisphosphate or 1 fructose 6 phosphate is the same, exactly same enzymes and reverse direction of the glycolysis pathway. And once it forms the fructose 1,6 bisphosphate, the reverse reaction from fructose 1,6 bisphosphate to fructose 6 phosphate is different. Here, the enzyme required is a fructose 1,6 bisphosphatase, where it will break down and gives the phosphate group. That's it. Not like glycolysis. Glycolysis, ATP utilizing, but here we cannot get the ATP from this reaction. Just inorganic phosphate is releasing, and the enzyme is different, that is fructose 1,6 bisphosphatase. Phosphatases are the enzymes will release the phosphate group from the substrate. Kinases are those, it will add the phosphate group. Okay. So the final one. So what is that? Glucose 6 phosphate to glucose. So in the reverse reaction in glycolysis, what happens? Glucose to glucose 6 phosphate catalyzing by the either glucokinase or hexokinase. But those two enzymes cannot revert back this reaction. So it required a special enzyme called glucose 6 phosphatase, where glucose 6 phosphate will be converted into glucose by releasing the phosphate moiety from the glucose 6 phosphate. And in the gluconeogenesis, then glucose 6 phosphatase is a hydrolyzed to glucose and inorganic phosphate, mainly occur in hepatocytes, that means in the liver cells. The glucose 6 phosphatase reaction allows the liver to supply the blood with free glucose. So with that, this glucose will be all produced glucose molecules in the liver will be supplied to the whole body through the blood, especially those cells needed like brain, RBC, testes, and uh, corneal cells. All those things mainly depend on the glucose on as energy. Okay, so this glucose 6 phosphate convert into glucose, then this glucose will be supplied through the blood. Okay, so that is how the pyruvate will be converting back to the glucose. So not only the pyruvate, right? So pyruvate synthesizing from lactate. So lactate reverts back to the pyruvate, lactate dehydrogenase, and then pyruvate will be converting into the glucose, all the steps we have just now seen. So thus, lactate also utilized in the gluconeogenesis. How? Lactate will be converted into pyruvate first, then pyruvate in the subsequent steps is formed in the glucose. And alanine also utilized as a part of the gluconeogenesis because alanine first undergo the amino transferase transferase reaction where amino group will be released from the alanine and it forms the pyruvate, and pyruvate undergo the steps which we have seen, then it will form the glucose. Okay, that is clear. Apart from that, glycerol also can convert into pyruvate. How? Glycerol will first convert into glycerol 3 phosphate by utilizing the ATP, and glycerol 3 phosphate convert into dihydroxyacetone phosphate, which is the intermediate of the glycolysis. Once it forms two molecules of dihydroxyacetone phosphate, then one of it will be converted into glycerol DH3 phosphate. Both will combine to form fructose 1,6 bisphosphate. Then further, the same thing which we have discussed just now will be converted into glucose. So what are the molecules utilized for the gluconeogenesis or for the glucose molecule synthesis? Pyruvate, lactate, alanine, glycerol. These are all the molecules can be utilized as a non-glucose source for the synthesizing glucose. These are all called as gluconeogenesis. Okay, so yesterday we were talking about the formation of the lactate, especially in the muscle cells when you are taking vigorous exercise continuously, when you don't have sufficient oxygen supply, which are need to be undergo all the pyruvates to the aerobic respiration, aerobic pathway. So then what happens? The deposited lactates in the muscle, it is going to be, it is accumulate, it's going to be toxic. So what happens? There is another possible way is present by the body. So what happens? This pyruvate, lactate from anaerobic glycolysis mainly in the contracting skeletal muscles and RBC carried out to the liver by the blood. So all will be collected by the blood and it will pass to the liver. And liver, what happens? Reversibly oxidized to pyruvate by lactate dehydrogenase. So what happens? The reverse reaction of the lactate into pyruvate happens in the liver cells. And pyruvate by gluconeogenesis in the liver synthesize the glucose. So pyruvate produced from the lactate, what happens? It undergo gluconeogenesis and will produce glucose. And then glucose releases from the liver to carry out the blood to meet the energy requirement by the peripheral tissues. So again, this glucose will be passed to the all the tissues. So this is called as Cori cycle. So when the peripheral tissues utilizing the glucose, forming the lactate will be passed to the liver by the blood, and in the liver, the lactate convert back to the pyruvate convert back to the glucose by gluconeogenesis. Then that resulted glucose will be again transported to the all the peripheral tissues through the blood, then glucose again utilized. So this is a cycle. Okay, so that is called as Cori cycle. So you can see this figure, it will illustrate clearly. Glucose it utilize in the glycolysis, and when it is good, the anaerobic glycolysis, so it will produce lactate in the muscles, and the lactate taken by the blood to pass to the liver. So the lactate undergo the gluconeogenesis through the pyruvate, it will form the glucose, and again glucose will be passed to the transported to the skeletal muscle through the blood circulation. So this is a cycle. Okay, this is called as Cori cycle. Just now I told you alanine also can give the no the pyruvate, and glycerol also can undergo gluconeogenesis, because glycerol will form glycerol 3 phosphate, then glycerol dihydroxyacetone phosphate, then so this is how glycerol will be glycerol 3 phosphate, then dihydroxyacetone phosphate, then it will convert into glycerol 3 phosphate, another molecule of this together fructose 6 1,6 bisphosphate, and so on, it will form the glucose. So glycerol also can be used in the formation of the glucose, called as gluconeogenesis pathway. And this is the regulation. Okay, maybe gluconeogenesis regulation we can talk in the next class because it is already